Yeah. So sorry for the delay. There was a confusion. Yeah. So now today we'll talk about virology. So what is virology? What are viruses? So virology is nothing but the study of viruses. What are viruses? They are the very primitive microorganisms, right? They're very primitive in the sense usually we divide the microorganisms into prokaryotes and eukaryotes, right? So basically viruses, where do they fall into? Viruses will not fall into either prokaryotes or into eukaryotes because they're very primitive that they do not fit into anything. So this is a very important MCQ that viruses are either prokaryotes or eukaryotes, then your answer would be none of it, right? So eukaryotes are the higher microorganisms, prokaryotes are lesser than eukaryotes, and viruses are very, very primitive that don't even fit into any of them, not even prokaryotes, not even into eukaryotes. Okay, so this is your first MCQ being discussed. So now, how do the viruses differ from bacteria? There are many properties of virus by which they are going to differ from viruses. What are they? We'll see them one by one. So one is viruses are obligate intracellular. That means they cannot survive without a cell. So they always will be able to survive only inside a cell. That is why we don't have anything like an artificial culture medium for growing viruses, but we have something called a cell culture. So that is different from the culture medium which we use for bacteria for the growth, right? So they are obligate intracellular, that is the first MCQ. And viruses, they possess either RNA or the DNA as the nucleic acid, but never both. But compared to the bacteria, they can have both. So viruses, they either possess RNA or the DNA, and they never possess both of them. And the next important point about viruses, they're very minute in size. So they are in nanometers. So what happens is the filters which are commonly used for bacterial sterilization, since the viruses are very, very, very small in size, they get filtered very easily. So they can pass through the filters that are available for the bacteria. So basically, viruses are filterable. They passes through the bacterial filters. The next point is, if you talk about the multiplication of viruses, the multiplication of virus is very complex. They multiply in a very complex way. Compared to the bacteria, they divide by means of binary fission. It's a simple division, right? But the multiplication of viruses, they are very complex. So this is one of the differences. And viruses lack the enzymes for the synthesis of proteins. So that is why they are always dependent on the host for their multiplication. So viruses lack the enzymes for protein synthesis. Okay, and viruses will not be susceptible to the antibiotics which are given to the bacterial infection, or the antibiotics which are active against the bacteria is not used for the viruses. For viruses, instead, we have separately the antiviral drugs. And as I told you, any virus, if you want to grow them, you cannot grow them in a cell-free medium. So viruses need live cells. They need the live cells for its growth and multiplication. So viruses cannot be grown outside a cell. Okay, so this is very important. And if you talk about viruses, they as such will lack a cellular organization compared to that of the bacteria. When we talk about bacteria have a proper cellular organization, right? They have a cell wall, they have a cell membrane, they have cellular organelles. But when you talk about viruses, they lack a proper cellular organization. Okay, and they also lack the cell wall and the other structures which are present in the bacteria, as we study in detail about the bacterial structure, as those structures of structured organization of virus is not there. Okay, so being said that, we'll move into the various viruses. Okay, so when we talk about this, so when we talk about this particular figure here, so what you're seeing here is, so on your right side, there is a figure which is showing there is a nucleic acid inside, followed by you are seeing surrounding structure that is called as a capsid here, right? So you have a nucleic acid surrounded by a capsid. So what is this? Nucleic acid is basically either RNA or a DNA, that is ribonucleic acid or deoxyribonucleic acid, and it is surrounded by the capsid. What are capsid? Capsid are basically nothing but they are the protein capsomeres. They are the protein structure which surrounds the nucleic acid. So this forms the whole thing together with the capsid along with the nucleic acid together called as a nucleocapsid. So it's very important when you talk about the basic structure of virus. The nucleus with the nucleic acid along with the capsid is called as a nucleocapsid, right? So this nucleocapsid, when you're talking about, depending on the structure, you see this structure here. So this structure is called as an icosahedral, right? Or when you see this type of structure, which is of a helical, then it is called as helical. So any virus, they can be either icosahedral or they can be a helical virus, right? The structure can be either icosahedral or it can be helical. So this is about the naked virus. That is, it has nucleic acid and the capsid. So there are viruses where it can be surrounded by an envelope. So when it is surrounded by an envelope, it will be called as an enveloped virus, right? So this enveloped virus, if you see here, it has a lipid layer, right? This particular thing is a lipid layer, and you can see some spikes over the envelope. Those are called as the peplomeres, and those are basically of glycoprotein. So the membrane, the envelope is made up of lipid as well as protein. So that is also is very important. So basically, when we talk about any virus, we will say either the nucleic acid is RNA or DNA, whether the structure is icosahedral or helical, whether the virus is enveloped or it is non-enveloped. So this is the basic thing about the structure of the virus. So here you are seeing a icosahedral naked virus. This is a helical naked virus. This is a helical enveloped virus, and on your rightmost corner, that is an enveloped icosahedral virus, right? So we'll move on to one by one discussion. So as I said, the virus structure can be either helical or it can be icosahedral, and the virus can be with or without the envelope. The nucleic acid can be either RNA or it can be DNA. And when we talk about the nucleic acid, there is one more MCQ of importance. The question can be asked as the genomic size of the largest virus and the smallest genomic size. So that would be the largest the largest genome is found in the retrovirus. So that is measuring around 7 to 11.5 kilobase pair, right? Whereas smallest will be hepatitis D virus, that is around 1.7 kb, followed by hepatitis B, that is 3.2. Suppose you have an MCQ asking which of the viruses the nucleic acid has the largest genome, then your answer will be retrovirus. If the question is the smallest, the virus which has the smallest genome, then your answer would be, if you have hepatitis D virus, then your answer would be hepatitis D. If there is no hepatitis D, then it would be hepatitis B virus, right? So this is another MCQ. And all the RNA virus has a single copy of unsegmented RNA virus. There are exceptions. One is a reovirus, which has double standard RNA virus, which has a double standard RNA genome. It's very, very important MCQ. The MCQ will be all has single standard RNA except. If you see that question without even looking at the option, the answer is the reovirus, because reovirus is the only RNA virus which has the double standard RNA. Okay. Then you might have a twist in the question, that is retrovirus. It has only a single standard RNA, but it has 2 copies of single standard RNA, right? So retrovirus, including HIV, has a double copies of single standard RNA. So that you should be aware about, right? So reovirus is the only RNA virus which has double standard RNA. Retrovirus has single standard, RNA, but it will have 2 copies of single stranded RNA. Very important, right? Yes. So we'll move on to the next questions. Right. So since said that about the RNA, the nucleic acid, you also please remember there are the RNA viruses which are segmented. Okay, segmented RNA. So remember the mnemonic ABIR. Okay, ABIR. That is in the sequence of segments I have arranged them. So that is arena virus have 2 segments, Bunia virus have 3 segments, influenza virus have 8 segments, and rotavirus have 11 segments. So this can be a very important MCQ. The MCQ can be all of them have segmented RNA except, right? If you have something else in the question, say you have hepatitis, or you say you have, sorry, these are about RNA virus. Say you have some other option, then you should know what are all the segmented RNA viruses. Please excuse me. Yeah. So remember this mnemonic. So what you can, how you can remember the number of segments? You remember it as 23rd of August 2011, right? So that will be 2, 3, 8, and 11. So the mnemonic will be ABIR. So that is arena virus, Bunia virus, influenza virus, rotavirus has 11 segments, influenza has 8 segments, and Bunia has 3 segments, and arena virus have 2 segments. Remember it in this sequence. It's an important MCQ. Now, when I talk about the segment enveloped virus, what is important about enveloped and a non-enveloped virus? When I talk about enveloped, I told you it is a made up of lipid, and also it has peplomers, that is of glycoprotein. So they are all the lipoprotein subunits, and they are called as the peplomers. So whenever an virus is enveloped, that means this particular virus has derived the envelope from the human membrane. So that is very important. And the protein part is always the virus derived. So when the virus derives its envelope, it is always from the host, and the protein part of the envelope is always the virus derived. So this is very important that you remember. Okay. And whenever any virus which is enveloped, it will be heat sensitive and it will also be ether sensitive. That means when an enveloped virus is get in contact with ether or with heat, it is going to be destroyed. So that is the meaning. Okay. So it is heat labile as well as it will be ether sensitive, the enveloped viruses. So basically, enveloped viruses are not very effective when there is heat or when there is a kind of ether or acid in the surrounding, then this virus is going to be get inactivated, right? So that is the meaning. So when I talk about the non-enveloped viruses, so I told you already, enveloped viruses, it will be just opposite to the enveloped viruses for non-enveloped viruses. So they will be ether resistant and it will also be heat stable. That means in a higher temperature, even they will remain stable. And one more important thing about the non-enveloped viruses is most of the viruses spreads through the gastrointestinal tract, that is through the fecal-oral route. Most of the viruses are non-enveloped virus. So it's a very, very important, point for you to remember the route of transmission of even diseases, because most of your viruses which are spread through the gastrointestinal tract, they are all non-enveloped viruses, because when the enveloped virus get inside your gastrointestinal tract, the acid is going to destroy the virus and it is going to lose its property of, infecting the cells. So most of your non-enveloped viruses are the one which is going to cause the infection through the gastrointestinal tract, right? Okay. So you also have to remember what are all the non-enveloped viruses. So remember, for non-enveloped viruses of DNA virus, remember the mnemonic as AP. Okay, AP. That is adeno, parvo, and papaver. So these are the 3 viruses which are non-enveloped DNA viruses. The non-enveloped DNA virus, remember the mnemonic AP, adeno, parvo, papa-O. Everything ends as O, O, O. Okay. So that is the non-enveloped DNA viruses. And the non-enveloped RNA viruses, you have picornavirus, calcivirus, and reovirus. So these are the non-enveloped RNA viruses, where most of the viruses like polio, ecovirus, enterovirus, rotavirus, all these are the RNA viruses which are non-enveloped viruses, right? Fine. So moving on to the DNA viruses in detail. You have to remember the list of DNA virus, because the question itself will be all are DNA viruses except. So now you should know what are the DNA viruses list. So for the DNA virus list, we have a mnemonic, and that is called as happy. Okay. Remember it as happy. That is the mnemonic for the DNA viruses. So HAPPY. So this is a mnemonic for RNA virus. So remember, there are 2H, 1A, and 3P for DNA viruses. So H is for herpiviridae. So I'm talking about the family as such. H is for herpiviridae. Another H is for hepadnaviridae. A is for adenoviridae. P is for poxviridae. P is for papoviridae, and other P is for parvoviridae. Okay. So being said that, so the mnemonic for DNA virus is happy. Okay. So there are 2H, 1A, and 3P. So remember this: herpesviridae, hepadnaviridae, adenoviridae, poxviridae, papoviridae, and parvoviridae are the DNA viruses. So now, what are the viruses in each family of viruses of DNA? When I talk about herpes viridae, the virus in herpiviridae are herpes simplex virus 1, herpes simplex virus 2, varicella zoster, that is also called as herpes simplex virus 3, then cytomegalovirus, Epstein-Barr virus, human herpes virus 6, 7, and 8. So all these comes under the family of herpiviridae, right? And what is the virus which comes under hepadnaviridae is hepatitis B virus. Hepadna, remember it is hepatitis B virus. Adenoviridae, we know adenovirus comes under adenoviridae. Poxviridae, we have the variola, vaccinia, and molluscum contagiosum virus, are the ones which comes in the poxviridae. Okay. So poxviridae, you have variola, vaccinia, and molluscum contagiosum virus. Then you have the papoviridae. So in papoviridae, you have the human papilloma virus, polyoma virus, BK, and JC viruses. Okay. So these are the viruses which comes under papoviridae. Human papilloma, polyoma, BK, and JC comes under the papoviridae family. Then parvoviridae. Parvoviridae, the virus which comes under parvoviridae is the parvo virus, right? So these are the important DNA viruses we are talking about. Remember, you have herpiviridae, hepadnaviridae, adenoviridae, poxviridae, papoviridae, and parvoviridae. So kindly remember this. It is a very, very important MCQ. So you should know the list of DNA viruses. Okay. So we'll go to them one by one. So when I talk about DNA viruses, most of the DNA virus has an icosahedral symmetry. Most of the DNA virus have icosahedral symmetry except for pox virus, which has a very complex symmetry, right? So this you should know, right? So we have the enveloped icosahedral DNA virus. If I talk about the enveloped, then we have hepadnaviridae and the herpes virus. Okay, hepadnaviridae, I told you it is hepatitis B virus, and herpes viridae, I told you we have the others, in the herpes virus, right? So as I have told you here, hepadnavirus, that is having hepatitis B, has a small genomic sequence size, genome size, that is around 3.2 kb. The smallest would be hepatitis D virus, right? Followed by hepatitis B virus. So hepatitis B virus and herpes simplex virus, they are enveloped DNA virus which has an icosahedral symmetry. Please remember it. Okay. So the next thing here, what you are seeing here is the list of the non-enveloped DNA virus, but they are of the icosahedral symmetry. One is adenovirus, parvovirus, polyoma virus, and the papilloma virus, right? So these are the non-enveloped DNA viruses which has the icosahedral symmetry, right? So this picture, if you could remember, you can remember the list of enveloped and non-enveloped DNA virus. As I've already told you, all the DNA viruses have an icosahedral symmetry except for a pox virus, which has a very complex symmetry, right? Okay, so we'll talk about the RNA viruses. When I talk about the RNA viruses, it can be either single standard or it can be double standard. When I talk about the single standard RNA virus, it can be either a positive sense RNA or it can be a negative sense RNA, right? So if I talk about the negative sense RNA, single standard negative sense RNA, all of them are enveloped. But when I talk about the single standard positive sense RNA, they can be either enveloped or they can be non-enveloped. And there is only one double standard RNA, and that is non-enveloped. Okay, there is only one double standard RNA that is also non-enveloped. So now we'll go on to one by one. So I'm talking about a single standard negative sense RNA, and all are enveloped. So the examples for single standard negative sense RNA, which are all enveloped, there are the orthomyxoviridae, paramyxoviridae, rhabdoviridae, bunyaviridae, arinaviridae, and filoviridae, right? So remember, it is myxovirus. So it is orthomyxo and paramyxo, which is of negative sense, and rhabdoviridae, right? Bunyaviridae, arina, and filoviridae. And these are the viruses which are negative sense single standard RNA, right? And then when we talk about the positive strand, enveloped, those are Togoviridae, Coronaviridae, which includes the coronavirus, Retroviridae, which includes HIV, and then you have the Flaviviridae. These are all the enveloped positive stranded single sense positive stranded enveloped RNA virus. Okay, TCRF, right? Togaviridae, Coronaviridae, Retroviridae, and Flaviviridae. And when we talk about the single standard positive non-enveloped, then it is Picornaviridae and Calciviridae, right? It is PC. The non-enveloped virus is single standard positive. It is Picornaviridae and Calciviridae. And we talk about the double standard non-enveloped RNA virus. It is the Reuviridae. Okay, please remember, it is the only RNA virus which is of the double standard RNA virus, that is the Reuviridae. So now we'll move on into the details so that you can remember it better. So here you can see that this particular viruses, I have put it in a red color box, right? And also I have put a cover over it. So this cover is going to tell you that it is enveloped, but I have given a red color to it. That means it is a negative sense RNA, which is enveloped. Okay. So remember, it's orthomyxo, paramyxo, rhabdo, bunya, arina, and filovirus, right? Orthomyxo, paramyxo, rhabdoviridae, bunyaviridae, arino, and philo. So you can make your own mnemonics to remember them, right? So mixo, remember it's mixo, right? So there is a mixing, right? So there is rhabdoviridae, bunyaviridae, arino, and filoviridae. So these are the RNA virus which are enveloped with a negative strand RNA. The next box which I have made it with a green color, and I have made a green color cover. That means they are the enveloped virus, but they have a positive strand RNA. So the examples are Togoviridae, Coronaviridae, Retroviridae, and Flaviviridae, right? So these are the positive sense RNA which are enveloped. Then I have made Picornaviridae and Calciviridae. So these are the non-enveloped positive strand RNA. So here I have again made it green, but I have not made it a cover. Just I have given a green outline to it. It is a positive strand, but it is non-enveloped. So that is Picorno and Calciviridae. The next thing is I have made Rio Viridae twice for you to remember that Rio Viridae is a double-stranded RNA virus, right? So this you have to remember. So we will move on to one by one. What are the viruses inside each of the family? When I talk about Orthomyxoviridae, you have to remember the most important virus, which also is known to cause the epidemics and also pandemics. So the virus is influenza, right? And the subtypes are influenza A, B, and C. Next, you have the Paramyxoviridae. In Paramyxoviridae, you have parainfluenza, mumps, measles, respiratory syncytial virus, and metapneumoviruses. So these are the virus which comes under the Paramyxoviridae. Rhabdoviridae, the name itself says rabies. Okay, rhabdo is rabies. Okay, so remember this also. Rabies is going to cause a high difficulty in the patient. So remember it is a negative impact. So remember it is a negative sense RNA. When I talk about negative sense RNA, all the negative sense RNA are enveloped. So remember this, it is an enveloped virus. Then you have Bunya virus. Bunya virus again, hantavirus and one of the hemorrhagic fever virus. Sandfly fever virus comes under Bunya viridae, and Arena viridae has lymphocytic choriomeningococcal virus, Lassa fever virus, right? And Filoviridae, again it's the hemorrhagic fever virus, that is Marburg Ebola. So Marburg Ebola, hemorrhagic fever, negative impact. So negative sense RNA, all the negative sense RNA viruses are enveloped viruses. Please remember, so Filoviridae, Marburg, and Ebola. So this particular slide, if you're thorough like this, that is why I made it easier for you to remember. Visually it is easy, like when you see it visually again and again, definitely you will be able to memorize it, right? Next is the positive strand single stranded RNA. So you have Togoviridae. In Togoviridae you have rubella, eastern equine encephalitis, western equine encephalitis, even chikungunya virus, which falls under the Togoviridae family. Then coronaviridae, we all know that coronavirus comes in the coronaviridae. Retroviridae, we have the HIV and HTLV. HIV is human immunodeficiency virus. HTLV is human T lymphocytotrophic virus. And flavivirus, flaviviridae is the most common viral diseases which are in India. That is, you have dengue and hepatitis C virus, West Nile fever virus, and yellow fever virus is not endemic in India. Okay, so yellow fever is not endemic in India; it's endemic in South Africa, right? So flavivirus, if I talk about, it is dengue, yellow fever, hepatitis C, and West Nile virus. And when I talk about picornavirus, I told you all the non-enveloped viruses are going to spread through the oral route. So if I talk about polio, fecal-orally transmitted, echo, fecal-orally transmitted, enteroviruses, fecal-orally transmitted, hepatitis A, fecal-orally transmitted, Coxsackie, rhinovirus, all these also can spread through the gastrointestinal tract. So picornaviridae have the most important viruses which are spread fecal-orally, that is polio, echovirus, entero, and hepatitis A viruses, which are of importance. Calcivirus, yes, hepatitis E virus. So please remember, when we talk about hepatitis virus, we have A, B, C, D, E, and we have E, right? So the A and E, when we talk about the hepatitis virus, B, C, and D, they are all transmitted through blood transfusion and also like the blood transfusion and sexually transmitted. But when we talk about hepatitis A and hepatitis E, they are the exceptions of the hepatitis virus that they spread through the fecal-oral route. So here remember that hepatitis A and hepatitis E, they spread by fecal-oral route. So remember they are all the non-enveloped and they are the RNA virus with the positive strand, right? So Reoviridae, again, it is the rotavirus. Where did we study rotavirus? Rotavirus we studied in a place where they are the viruses without envelope and they are also the segmented RNA viruses, right? So Reoviridae, you have rotavirus. Now, we have seen the classification of viruses, DNA and RNA virus. Now, we should also know about the viral replication. As I told you, the viral replication is very complex compared to the bacteria and other organisms. So, replication of virus has to need to undergo many steps. So, they are. So, as I have shown you the picture here, that is the first step is either the attachment or the absorption of the virus to the cell, right, on the human cell. It has to attach or the absorption has to happen So there must be the human host cell receptor and the virus interaction happens. So there is this virus receptor and virus interaction. So they adsorb to the surface of the host cell. Then comes the penetration. So the penetration can either happen through the endocytosis, whether the cell itself can phagocytose the virus, that is phagocytosis, or it can happen as a membrane fusion, and as it happens in HIV, in human immunodeficiency virus, the virus will penetrate the host cell membrane by means of fusion, right? It penetrates by means of the fusion. Or it can be just the injection of nucleic acid, which is done by means of the bacteriophages. The bacteriophages, they just inject the nucleic acid into the host cell. They actually do not enter the host cell, right? So that is injection of nucleic acid as happening in the bacteriophages, right? So this is penetration. Once it penetrates inside the human cell, it has to uncoat, right? The nucleic capsid and the nucleic acid has to uncoat for it to replicate. So uncoating step will happen, and then it leads on the next step would be the biosynthesis. Whatever enzymes and proteins that is required for the biosynthesis, as I already told you, they lack the enzymes required for the biosynthesis. They are always dependent on the human cell for the replication. Okay, so if I talk about biosynthesis, remember this is a very important MCQ and also for your exams. The replication for the virus, it either happens in the nucleus or it happens in the cytoplasm. So when I talk about this, so the replication of the DNA virus, remember D as Don. Don is a very important person, right? He always occupies a center position. So remember, the DNA viruses, mostly the replication D for Don, they happens in the nucleus. There is only one exception, that's pox virus, right? Where it happens in the cytoplasm. So D for Don, that is the center. So nucleic acid, so nucleus. So DNA replication happens in the nucleus of the host cell, right? Except for pox virus, where the replication happens in the cytoplasm, right? So RNA, remember R for run, right? You are supposed to just run away, right? So that it will be easy if you are in the cytoplasm, it will be easy for you to just run away. So R for run, remember all the RNA viruses, the replication happens in the cytoplasm, except for retrovirus and the orthomyxovirus, where it happens in the nucleus, right? So RNA, the biosynthesis happens in the cytoplasm, except in retrovirus and in the orthomyxoviruses. Next thing is assembly. So once all their nucleic acid and everything is formed, the virus has to finally assemble for it to form a complete viral structure, right? So assembly again, as I already told you, the DNA virus is going to biosynthesis is going to happen in the nucleus, right? So the assembly also will happen in the nucleus. Only exception is the Hepidna virus and pox virus. Pox is already, the biosynthesis itself is in the cytoplasm. So the assembly also will be in the cytoplasm. But the Hepidna virus, the synthesis will be in the nucleus, but when it goes for the assembly, it happens in the cytoplasm. Similarly, all the RNA viruses, I have already told you, the assembly is going to be in the cytoplasm itself, right? Whereas the synthesis of all the RNAs in the cytoplasm, except for retrovirus and the orthomyxovirus, but the assembly of all the RNA viruses is going to be in the cytoplasm. And then maturation. Finally, they are going to mature into a proper virus structure, and then they are going to come outside. When I talk about the release of the virus can either happen because it is happening inside, the whole cycle is happening inside the cell. So the release can only happen when it has to go out of a cell. So this release can happen when the cell itself is going to lyse, right? The cell is going to lyse and then it's going to release the viruses. In that case, when it is going to just release out of lysis of the cell, the virus will not take up the envelope of the cell membrane of human beings. So they all come out to be the non-enveloped viruses, right? But when some virus which are going to bud out, when they're just going to bud out of the host cell membrane, they will carry the envelope. They are going to carry the cell membrane of the host cell, and they all become the enveloped viruses. Okay, so this is about the viral replication. I'm sure everybody is interestedly noting down all the points, and you will get the maximum benefit if you are just going to follow this PPT, because this presentation in the general virology, you'll be able to answer 100 out of 100. And in the meanwhile, I also request you to kindly subscribe to the Nextillo app, and please download and get the maximum benefit. Okay. So since that again, the same point I have already told you, all the RNA viruses, sorry, all the DNA viruses, the replication is going to happen in the nucleus, except for pox virus, where the replication is in the cytoplasm. And all the RNA viruses, I told you, you are just going to run away, run R. So that is in the cytoplasm, except for the influenza virus and the HIV virus, which is going to happen in the nucleus. So here, yeah, this is a very, very, very important MCQ. All the DNA viruses have a double stranded DNA except for parvovirus, which has a single stranded DNA virus. So parvovirus has a single strand of DNA. It's a very, very important MCQ. And all the RNA virus has single stranded except for reovirus, which is having a double stranded RNA virus. I told you already, this question has been discussed already, right? Fine. Now then, there is something called as an eclipse phase. What is an eclipse phase? See, suppose I got a viral infection. So a virus entered into my body, but it will take some time the virus to go and replicate and, to produce its progeny and to bring it out in the host. So that particular time period is called as the eclipse phase. What is it? It is an interval between the entry of the virus until the appearance of the infectious particle, that is infectious viral particle. So this time gap is called as the eclipse phase. And in this eclipse phase, you cannot demonstrate any virus or any, during this period, you cannot demonstrate any of the virus, like the protein of the virus or antibodies developed to the virus. You cannot demonstrate in the host. So that is called as an eclipse phase. Being said that, this figure I just wanted to show you. You see the size difference on here. So this is the pox virus. I told you it has a complex symmetry, right? So pox virus also, the shape here, you can remember it has a brick shaped. It's a brick shaped virus. That's pox virus. You see, this is the largest virus, and compared to here, parvovirus is the smallest virus. And parvovirus is also, it has a single standard DNA virus. So this is a very, very important point you have to remember, right? So pox virus is a brick shaped virus I want to show you here. And here you can see adenovirus here. This is a adenovirus. The shape of adenovirus is described as space vehicle shaped. Very, very important question. Okay, it is a space vehicle shaped virus as for the adenovirus. And parvovirus, I have already told you. So these are the different DNA viruses. Now the next one, next picture. Yeah. So this picture, I just wanted to show you the different shapes of the RNA virus, right? So here you can see the RNA virus. When I talk about, few important things which you have to remember are, one is, here you can see here, this is the coronavirus, which has these sun ray appearance, right? So they have these peplomers, which is on the surface, and it gives a sun ray appearance to the virus, corona shape, right? Sun ray appearance to the virus. So that is the coronavirus. And then I also wanted to show you, this is, you can see here, the filovirus, which has this filamentous structure, right? Which has the filamentous structure. When we talk about filovirus, I told you filovirus, the viruses which come under filovirus are Marburg and Ebola. So they have a very filamentous structure of the filamentous RNA structure. And here I also want to tell you about, very particularly about this rhabdovirus, that is the rabies which comes under it. What is very peculiar and very important about rhabdovirus is it has something very peculiar. It's called as the bullet-shaped virus. See this? Okay, it is a bullet-shaped virus. Okay, it is a bullet-shaped virus. It is also called bullet-shaped virus, that is rhabdovirus. So being said that, we will move on to the next slide. So now I told you that virus cannot be cultured in the artificial culture medium, except that when there is a live cell. When you have a live cell, you will be able to, grow the viruses inside the cell. So in vitro, that means without any living cell, you can never grow the viruses. Because I told you already, because they lack the enzymes necessary for multiplication, they are always intracellular. They need the host cell for their replication. So they cannot live without any cell. So the viruses always needs cell for its survival and replication, right? How do you grow the viruses. If at all you want to grow it, how do you grow it? So one is like you can put it or grow it in the animal, right? You can grow them in the animal. So what animal is commonly used? The various animals which are being used, but most commonly you can use that something called as suckling mice, right? So this is a suckling mice which I've shown you here. So suckling mice is anything which is of, the age is only around 1 to 7 days old. So it is in the suckling phase. So those mice are called as a suckling mice, and this suckling mice is commonly used for the growth of animals and also to check about the pathogenicity and also about its virulence. So we use mice for the animal studies. So the suckling mice, when we talk about the animal inoculation, you have something called as uh, the most common virus which are used to grow the viruses using suckling mice is Coxsackie A and Coxsackie B viruses. Coxsackie viruses are the most commonly used, most common viruses that is being grown in the suckling mice. So if when I talk about Coxsackie, you have Coxsackie A and B. When you inoculate this virus into the mice, so it's going to cause a flaccid and a spastic paralysis. So remember, A comes first, B comes later, F comes first, S comes later, right? So Coxsackie A is going to cause a flaccid paralysis. Coxsackie B is going to cause a spastic paralysis in the mice, right? So the other medium which is used, the other place where you can grow the virus is the embryonated egg inoculation. Embryonated egg is different from the eggs which you get it in the shops, right? Those are all the non-embryonated eggs. So embryonated eggs are specifically, they're all brought in the labs, right? So embryonated eggs are the ones with the embryo, right? So those eggs will be like maintained in the laboratory for a period so that the embryo grows in a proper way so that the viruses can be grown in the embryo inside the egg. When I talk about the embryo, yeah. So the other method to grow the virus is the tissue culture. There are three methods by which you can grow the virus. One is animal, one is embryonated hens, one embryonated egg, and the next thing is the tissue culture. When I talk about the embryonated egg, yeah, it's a very important MCQ. When I talk about the embryonated egg, you have four layers or the four places. One is the chorioallantoic membrane. The next would be the allantoic cavity. The next would be the yolk sac, and the next would be the amniotic inoculation, amniotic cavity. The four places where you can actually grow the virus. When you see this picture itself, you will be knowing the one here, that's the allantoic cavity, is the biggest of all the cavities. So this is the cavity where you have maximum space in the egg. Why I am telling you this point is because allantoic cavity is the place where the researchers will grow the virus for vaccine production. Okay, so allantoic cavity is a cavity which is used to produce vaccine. The very important question: vaccine production. You should remember the allantoic cavity because allantoic cavity is the biggest of all of them. So the maximum number of space availability. So that allantoic cavity is used for the vaccine production. Mostly it's used for influenza vaccine production. You use the allantoic cavity, right? When you talk about the different other places where the virus can be like grown, one is the chorioallantoic membrane. When I talk about chorioallantoic membrane, you're just going to see the growth in the membrane. So the growth in the membrane, they're also called as pox, P-O-C-K-S, the pox. Okay, when the pox are formed, depending upon the nature of the pox. So I could also say this is one of the cytopathogenic effects of the viruses which are growing in the chorioallantoic membrane. So basically, what all viruses can be grown in the chorioallantoic membrane? It is the herpes virus, pox virus, and rosarcoma virus can be grown in the chorioallantoic membrane. And in the amniotic cavity, we can grow influenza virus and mumps virus. And in the yolk sac, here I have written herpes simplex, but more importantly, you can also grow rickettsia, chlamydia, and Haemophilus ducrei in yolk sac. Okay, it is a very important MCQ. So you can grow herpes simplex, chlamydia, rickettsia, and also Haemophilus ducrei in the yolk sac. Then comes your allantoic cavity. Allantoic cavity, I already told you. The main important use of allantoic cavity is for the vaccine production, and also the most important vaccine production or virus used is for the influenza virus. So as well, you can grow the virus. They are the influenza virus, mumps virus, and the other avian adenoviruses can be grown in the allantoic cavity. Okay, it is a very important MCQ. It can also be asked in your MBBS curriculum exams, right? So the next question I'll move on to Tissue culture. What is tissue culture? You're going to take the live cells. You're going to take the live tissue, and in the live tissue, you have the cells. In those cells, you're going to grow the virus. Right? If you talk about the tissue culture, even in the primitive methods, like where they didn't had proper facilities where they can separate the cells, so they just started using the organ directly. When they started using the organ directly, that is called as the organ culture, part of the organ. And explant culture is nothing but a tissue is being minced, and these tissue which are being separated into tiny pieces, and those cultures are used, and that is called as an explant culture. And then comes the newest of everything, that is the cell lines. What are cell lines? Cell lines are basically nothing but the individual cells from a tissue is separated, and then these cells are grown in the medium which is going to have the tissue culture medium, and in that medium you're going to grow these cells, right? When these cells are there in the tissue culture medium, it is going to keep on multiplying. When they have the enough nutrition in the cell, when they have enough nutrition in the medium, they will keep on multiplying. So these are the individual cells which are allowed to grow, allowed to multiply, right? So they keep on, like, they will be living in the cell lines. So when I talk about cell lines, so this is a picture I would like to show you. This is the container which is used for the cell line. So this can be one of the MCQs. This can be a spotter in your exam. So this is the cell culture vial, we can say, right? Used for the cell cultures, right? When I talk about cell lines, the cell lines are also, they are basically divided into three types as primary cell line, right? What are primary? They are the primary cell lines which were discovered. That means these cells, they can only go up to 5 to 10 divisions, not more than that, okay? So they are the primary cell lines. The example for the primary cell line is the rhesus monkey cell line is one of the examples of primary cell line. And human amniotic cell line and chick embryo fibroblast cell lines are the primary cell lines where they have only limited divisions. The cell can go up to 5 to 10 divisions. Secondary cell lines, they can go up to 10 to 50 divisions. So examples are human fibroblast cell line that is used for cytomegalovirus, and MRC5 and W38. These are the cell lines that are used. They are the human embryonic lung cell lines. And the continuous cell lines are nothing but they are the cells which are derived from the cancerous cells. So they have the indefinite division. They can keep on dividing. So what you do is you have to, maintain those cell lines in the cell culture. So they will keep on in the medium. Whenever there is energy, they will keep on multiplying and they will stay alive, right? So repeated passages. So you will be able to maintain these cell lines, and they are called as the continuous cell lines. And some of the cell line example is HeLa, that is human carcinoma cell line. HeLa is the cell line has got the name from the patient. So HE and LA are the first words of her name. So you got the name from the patient. Then HEP2 cell line, that is human epithelioma of larynx. KB cell line, that is human carcinoma of nasopharynx cell line. Vero, that is vervet monkey cell kid line, that is for rabies vaccine. Then BHK is baby hamster kidney cell line. Detroit is the sternal marrow cell line. McCoy is the human synovial carcinoma cell line, and that is used for chlamydia mostly. And you and you have the Chang CILNK lines, that is human conjunctiva, intestine, lung, and liver and the kidney cell lines. Okay, these are the various continuous cell lines. Why I'm telling you so much? There can be a question asked saying these are the continuous cell lines except. So you should know the list of cell lines. Okay, so being said that, suppose I am growing the virus. So I have a sample, I have put it in the cell line. I just want to know whether this virus is alive, whether the virus is growing, whether the sample is positive for the virus. How do you basically look for it? Okay, how do you look for it? That is called as the cytopathic effect. What is cytopathic effect? Whenever there is a virus which is growing in the cell, it is going to bring about some changes in the virus. Okay, so those changes are called as the cytopathic effects in the cell lines, right? So how are you going to see those cytopathic effects, or what are the cytopathic effects? One is, example is enterovirus, where it causes the rapid crenation and degeneration of the cells. So that is characteristic of enterovirus. And syncytium or a multinucleated giant cell formation. That is, all the cells will clump together to form a giant cell. That is very typical of measles and herpes simplex virus. Okay, this is the example where you can see the cell lines where the cells are all combined together to form the multinucleated giant cell. And then you have the rounding and ballooning of the cell lines. So that is also an important example is human herpes virus. So you can see here the cells have become rounding and ballooning. Yeah, the cells. So this is example is human herpes virus. Then you have cytoplasmic vacuolation. That is a property of simian vacuolating virus. And then when you grow adenovirus in the cell culture, they can attach one cell to the another, forming bunch of grapes-like structure. And that is the characteristic of adenovirus. Okay, this is a very important MCQ. This is called as cytopathic effect, right? And also how there is something called as viral interference. The other property, one is cytopathic effect. The other property of virus, whenever they grow in the cell line, is viral interference. What is it? Suppose there is a virus which can cause cytopathic effect, and there is a virus which does not cause any cytopathic effect, but it can still grow in the cell culture medium. So whenever you mix a cytopathic effect causing virus and a non-cytopathic effect causing virus into a cell line, this cytopathic effect causing virus cytopathic effect will be inhibited by the growth of non-cytopathic virus. So this is called as the interference. So the interference of a non-cytopathic virus is going to interfere with the cytopathic effects of the virus, which can cause the cytopathic effect. Okay, so this is an important question, that is interference. Okay, and detection of viral antigen in the cell culture. You can do a direct immunofluorescence and you can detect the viral antigen. And by electron microscopy, you can see the virus directly. I told you the viruses, the size of viruses are in nanometer compared to that of your bacteria, which is in microns. Microns is around 10 to the power of minus 6, whereas nanometers is in 10 to the power of minus 9. So to visualize a virus, you can go under the electron microscope, or you can directly detect the viral genes by means of antigen testing, or you can also go for polymerase chain reaction by PCR. You can detect the viral genes. And then also, whenever the virus is multiplying inside the cell, right, either the virus or the viral products, the viral proteins will leave behind something, some effect, and that's called as the inclusion bodies. Okay, when I talk about inclusion body, the inclusion body can be intracytoplasmic or it can be intranuclear. So when I talk about intracytoplasmic inclusion bodies, the most known and a common example is intracytoplasmic is Negri bodies that is seen in rabies. Okay, remember Negri rabies, right? Intracytoplasmic. Passion bodies is seen in variola. Guarneri bodies is seen in vaccinia. Bollinger bodies seen in foulpox, and molluscum contagiosum bodies are seen in molluscum contagiosum. So this is an example of, these are the ones which has the intracytoplasmic inclusion bodies. What is intranuclear inclusion bodies? When we talk about intranuclear inclusion bodies, they are divided into Cowdry A and Cowdry B type. So remember, there is a mnemonic HAPY, right? I'll show you the next one, then you will remember. So here you can see there is HAPY. Okay, so remember HAPY. So the H and the Y, they are the H and the Y are the Cowdry A type. The one in between, that is the A and P are the Cowdry B type. Okay, so remember HAPY, H and Y are Cowdry A, that is herpes and yellow fever. And in between A and P are the Cowdry B type inclusion bodies. So this is another MCQ. So the inclusion body of herpes is called as Lipschutz bodies, and the yellow fever inclusion bodies are called as Torres bodies. It's a very, very important MCQ. Torres bodies are seen in yellow fever. Lipschutz bodies, inclusion bodies are seen in herpes, right? So please remember this. And there are some of the viruses where they can have both intranuclear and intracytoplasmic inclusion bodies. The example, the first is measles. Measles has Warthin-Finkedley bodies. The inclusion body of measles is called as the Warthin-Finkedley bodies, and cytomegalovirus has an inclusion body that's called as owl eye inclusion body. The inclusion body of cytomegalovirus is called as owl-eye inclusion bodies. Please remember, okay? Cytomegalovirus. So this is the picture I'm showing here. So this is a picture. It shows you the intracytoplasmic inclusion body, right? And this is nothing but an example of a negri body, and the staining used for rabies negri bodies is called as stellar stain. Okay, stellar stain. Okay, and then next picture here, you can see this is the picture of both intranuclear and intracytoplasmic inclusion, and this looks like an owl's eye. So that is for cytomegalovirus, right? Remember, owl's eye inclusion bodies are for cytomegalovirus. Okay, being said that viruses infecting the human, they can be either treated or it can remain persistent for a longer time. So they can remain as a latent infection, but periodically it can exacerbate. Latent means the infection is still there, but it doesn't show its manifestation. Only sometimes it shows the manifestation. That's called as a latent infection. Then the cell can transform. There are some viruses where are oncogenic potential, like papilloma virus, hepatitis virus, right? They are all having the oncogenic potential. They can change cell transformation for oncogenicity. And then latency as seen in HIV virus, because the cell virus comes inside, it gets fused to the cell, and then sometimes it may not show the disease manifestation immediately. So there may be a latency. Or latency shown in some of the slow viral disease, that is nothing but the prion diseases, because these prion diseases are nothing but the incomplete proteins. So it also takes a lot of time for the disease to show its property. Or it can be just a persistent tolerant infection. That means the virus is still there, but the body becomes tolerant to the virus. The immune system does not fight against it anymore. That is called as a persistent tolerant infection. Said that we have something called as interferons. What are interferons? They are nothing but the cytokines that are produced by the host cell. By the induction of either a viral or a non-viral inducer, the body produces cytokines in response. Those are called as the interferons. So interferons, there are alpha, beta, and gamma. What is the mechanism of action? The interferons does not have any direct effect on the virus as such. Please remember, the interferons do not have any effect on the virus as such. But what it does is, the interferons goes and act on the host cells to produce something called as translation inhibitory proteins. And these translation inhibitory proteins are going to act on the messenger RNA, and then it's going to stop the translation of the viral RNA selectively, but not the human RNA, right? They will not stop the replication of human cells, but only the viral viruses, right? So these transcription proteins, translation inhibition proteins are going to selectively inhibit the proteins inhibition of the virus. So those are the interferons. Interferons are host specific, so they are not virus specific. As I told you, they do not have any direct effect on the virus, so they are not virus specific. The interferon synthesis begins immediately within hours of induction and reaches the high level within 6 to 12 hours. A very important point about interferon is interferons are not antigenic. They are poorly antigenic, so they're never used for the serological assay, right? So they're not used for any diagnosis, right? Okay. Said that the small last topic of discussion of today, that is the vaccines of viral infection. So when we talk about the vaccines, you should remember the vaccines of viruses. If I talk about the viruses, the vaccine can be either live attenuated, only the potential, the pathogenicity, the virulence is put down, can be either live attenuated, or it can be killed, or it can be a subunit vaccine, or it can be some of the diseases as both the live and killed vaccines for the diseases. So let's do one by one. Inactivated vaccine, we have rabies neural vaccine. That is, remember, there is a simple, there is a BPL vaccine and an infant mouse brain vaccine. The simple vaccine is nothing but phenol inactivated, but it is done in the sheep brain. BPL is the beta propanolactone inactivated, an infant mouse brain vaccine for rabies. And there are non-neuronal vaccines of rabies that is done in the cell culture. And the Caseinour forest disease vaccine, KFD vaccine, which is a killed vaccine. So in killed, you have only killed vaccine if you talk about, that is rabies and Caseinour forest disease. Caseinour forest disease and rabies virus as only killed vaccines. And live attenuated, you have mumps, that is Gerald Lyon strain, measles, Edmond Jagreb strain, rubella, RA273 strain, chickenpox, Oka strain of varicella zoster, smallpox, rotavirus, adenovirus, they're all live attenuated viruses. Remember, measles, mumps, rubella, chickenpox, smallpox, everything comes as the same, right? They're all the diseases which causes the rashes. Measles, mumps, rubella, chickenpox, smallpox, all these are having live attenuated vaccine. Then comes your rotavirus and adenovirus. Subunit vaccine, there are two you have to remember, that is genetically engineered vaccine, right? One is hepatitis B and one is a human papilloma virus vaccine. They are the subunit vaccines, okay? And then both, some of the viruses have both the live and killed vaccines, right? So polio has a oral polio vaccine, and that is the oral polio vaccine is a live vaccine, and that is OPV is live, and IPV injectable is a killed vaccine. Japanese encephalitis, you have Nakayama and Beijing strain, which is the killed vaccine, whereas SA14-14-2 vaccine for Japanese encephalitis, that is a live vaccine. Then you have influenza. The killed vaccine is given as injectable, and the live vaccine of influenza is given intranasal. Yellow fever vaccine, you have 17D vaccine, that is the commonly used vaccine, and that is live vaccine, live attenuated, whereas you have killed vaccines also for yellow fever. Then hepatitis A has killed as well as live vaccines. Okay. The next one is, yeah. So now that we have completed the general virology session, so I'll have few questions to ask you. We'll go one by one. Please identify this. Okay. So what is this? So if you have like listened to me carefully, I have told you this structure here you are seeing is showing something called as a filamentous structure, right? This filamentous structure, where it could be? Can it be coronavirus? No, it cannot be coronavirus, because coronavirus has structure. I told you already, it has a sundry appearance because it has the peplomeres in the envelope, right? So it's not coronavirus. Can it be Ebola virus? Can it be Ebola? What about HIV? HIV is definitely not this shape, and hepatitis also, it is not there. So the answer here is this particular structure is Ebola virus. Yes, the correct answer is Ebola virus. Okay. So identify this, the condition or the virus associated with this, or the virus which has caused this particular picture, right? Here, what are you seeing? Here you can see 1, 2, 3, 4 nucleus together combined. So when there are 4 nucleus combined together, that means there are 4 cells have combined together, right? That means they are the multinucleated cells. So that is multinucleated giant cell. Okay, this picture is a multinucleated giant cell. Will rabies cause it? No. Rabies we have already seen. Rabies has in the inclusion body that's called as Negri body, and that's not the picture. Cytomegalovirus we have already seen. It has an owl-eye appearance. That's not the picture. Adenovirus causes a cytopathic effect, that is the grape-like clusters in the cell culture, cell lines. That is not the picture. So this is an example of herpes simplex virus. So this is a zinc smear that is made from the lesion of herpes virus. So that is basically the multinucleated giant cell caused by herpes simplex virus. Okay. Okay. Question number 3, identify it. What is it? I have already told you this. This is a space vehicle appearance. Is it pox virus? Definitely not, because pox have a brick shaped appearance. Is it rabies? Definitely not, because rabies, I told you, it has a bullet shaped appearance. Is it a bacteriophage? No. Bacteriophage has a tadpole appearance, so it is not a bacteriophage. Okay, so basically this is an adenovirus which has a spaceship appearance, space vehicle appearance. So the answer is adenovirus. Okay. I'm so sorry, the answer itself is displayed. Okay, so all are live vaccine except, right? So rabies, KFD, injectable polio, and yellow fever 17D vaccine. See here, rabies, I told you, they don't have a live vaccine, right? So rabies is a killed vaccine. Occasional forest disease, I told you, it only has a killed vaccine. So again, it's not a live vaccine. So I said in polio, you have live as well as killed vaccine. So the live vaccine is OPV, the killed vaccine is IPV. So again, IPV is not a live vaccine. And then you have this yellow fever 17D vaccine. I told you the yellow fever has both live and killed vaccine, but yellow fever 17D vaccine is a live attenuated vaccine, and you have a separate killed vaccine. The answer here is all the A, B, C, or the killed vaccine. The answer for this question is the yellow fever 17D vaccine, which is a live vaccine. Okay. Question number 5. All are enveloped virus except, yeah, it's a very interesting question. I told you, please remember, non-enveloped virus is going to cause the disease which are going to spread by fecal-oral route. So anything which you see that is going to spread by fecal-oral route, that would be the answer, right? Fine. All are enveloped except, okay. The first thing is rotavirus. Influenza virus, it causes a respiratory illness. Rabies virus is spread through the bite of the rabid dogs. Coronavirus is again a respiratory disease. So influenza virus, rabies virus, coronavirus are all enveloped viruses. But rotavirus is a non-enveloped virus, and it is a virus which causes diarrheal disease in infants. So rotavirus is also, if you remember, they have 11 segmented RNA as the genome, right? So it's RNA virus, it is non-enveloped virus and also has a segmented RNA. So now we will, so the answer here is the rotavirus. Now we will move on to the question of the day. Question of the day. Yeah, so I would ask the students to answer this question, and the winner would be announced in the social platform of Nextillo. So please, try to answer. The student who answers first will get the award. Okay, the question of the day is, cytomegalovirus has the following type of inclusion body. Is it intranuclear, or is it intracytoplasmic, or is it both intracytoplasmic and intranuclear, or it is none? It's a very simple question I've asked you because I just want to make the questions more interesting. So I just asked you, is it intranuclear, or is it intracytoplasmic, or it's both, or it is none? So I'll give you like 2 minutes time so you can answer your question, like, right? So as you remember, I told you cytomegalovirus has an owl-eye inclusion body. I have already shown you the picture as well. I have explained you how the inclusion body would appear in the picture, right? I hope that everybody has answered, right? So here the answer for this question is, so now in this place, you should also remember the list of organisms which has intranuclear. Remember the mnemonic for intranuclear, happy. The H and Y are Crowdry A, and A and P are the Crowdry B inclusion bodies. And intracytoplasmic, I told you the list of organisms which cause intracytoplasmic inclusion. So, and also the list of organisms which has both the intracytoplasmic and intranuclear inclusion bodies, right? So, being said that, I think you must have answered this question. So the answer for this question is, cytomegalovirus has both intracytoplasmic and intranuclear inclusion bodies, right? Yeah. So here is the picture. I should told you already, it has an owl eye. It is a very typical owl eye appearance, right? It has both intranucleus and intracytoplasmic inclusion. Hope all of you have answered. And again, please subscribe to the Nextillo app and get the maximum benefit. And I would also like to say that this app is an exclusively having the educators of their own branch. So you will get the maximum benefit if you're going to learn from this app. So yeah, and as I already told you, virology is the neglected topic in microbiology. So I would make sure that in virology, you would be able to answer 100% of the questions. So being finished the first chapter of virology, we'll meet in the next session with some other viruses. And thank you so much. Have a good day. Thank you all.